Mycoplasmas pose a serious problem for anyone doing mammalian cell culture, and with potentially 35% or more of cell lines contaminated worldwide, it is critically important that anyone working with mammalian cells test their cultures on a routine basis. Invivogen has created a revolutionary assay named Plasmo test that allows anyone who is engaged in mammalian cell culture to test their cells in their own lab using basic cell culture techniques without the need for any special equipment. In this video, we will demonstrate the basic procedure of the Plasmo test assay. The Plasmo test kit will arrive with the HEC Blue 2 cells in dry ice or in a shipping flask if the order is shipped internationally. The remainder of the reagents will be shipped at room temperature and should be transferred to the appropriate storage temperature upon receipt. For maximum viability of the HEC Blue 2 cells, these should be thawed and grown immediately upon receipt. Before any mycoplasma detection assays are done, the HEC Blue 2 cells should be expanded and frozen stocks prepared. Once the HEC Blue 2 cells have been expanded, the mycoplasma detection assay can now be performed. The first step is to collect samples from the cell lines to be tested. Collect 500 microliters of supernatant. As there are some intracellular forms of mycoplasma, it is also important to scrape the bottom of the flask with the pipette tip to collect some adherent cells or homogenized suspended cells before collecting the supernatant. Heat the samples for 15 minutes at 100 degrees centigrade. This step is to ensure that native alkaline phosphatase are eliminated to eliminate false positives and to release any intracellular mycoplasmas that may be present within the cells. Let samples cool at room temperature. Samples can be tested immediately or stored at 4 degrees centigrade for up to 2 weeks. Prepare a 96 well plate for the assay, clearly labeling which wells will receive samples, positive or negative controls. Add 50 microliters of each sample and controls to wells in the predetermined layout. Now it is time to prepare the suspension of the HEC Blue 2 cells in HEC Blue detection. First, remove the growth media and rinse the HEC Blue 2 cells with PBS. Remove PBS. Add just enough fresh PBS to cover the bottom of the flask and detach the cells using a cell scraper. Trypsin should not be used to detach the cells, as this can activate the SEEP expression, leading to a high assay background or false positive results. Add 4 to 5 milliliters of pre-warmed HEC Blue detection media and gently pipette the cells to homogenize the cell suspension. Count the cells using a counting cell or automated cell counter. Prepare a cell suspension at 250,000 cells per mil in pre-warmed HEC Blue detection medium. Mix this suspension by gently pipetting several times. Add 200 microliters of cell suspension to each well containing samples or controls. Pipette gently a few times to mix cells and samples. Add cells to one empty well as a second negative control. Incubate the plate at 37 degrees centigrade with 5% carbon dioxide overnight. Visually inspect the plate for color change. Wells with the positive control should be dark purple or blue. Wells with negative control or cell suspension only should be pink. Read the cells in a plate reader at 620 to 655 nanometers. Any well with an OD of at least 3 times the negative control well, or a difference in OD of at least 0.5, is considered a positive result. For more information, please visit invivogen.com/plasmotest. If you have additional questions about this kit, please feel free to contact Invivogen using the information provided here.